Okay, welcome everyone. It's fantastic to have you all. Thank you very much for joining our closing event for the SusFE project. So we'll perhaps just give 1 or 2 minutes for other colleagues to join, just in case they're running a little bit late. So SusFE is part of a family of projects that was funded through the Horizon Europe program, supported through the European Commission. So we're really delighted to be able to present some of the outcomes from the project. Okay, so let's just give 1 or 2 further minutes to that, and then we can make a start. We'll have a number of really great presenters representing different partners, the different use cases that were developed within the SusFE projects, and they'll just introduce themselves as they do their presentations. I should introduce myself. I'm Zulf Ali. I'm the technical manager for the SusFE project. Also, in terms of my day job, I'm the Pro Vice Chancellor for Research and Knowledge Exchange at the University of Cumbria, and we've been involved in different elements of the work here. So what is the SusFE project about? As the title says, next generation sustainable and functional electronic components and systems. And it's about developing functional electronics for green and circular economy with roll-to-roll manufacturing, specifically for wearable and diagnostic devices. So there were a number of novel elements. There was a flexible integrated circuits, Flex IC, on polymer substrates, and that's about demonstrating particular electronic functions. A lot of that work was from a really fantastic company, Pragmatic. There's other work around use of a recyclable bioenzymatic fuel cell as a power source. There was some really interesting work that we'd introduce around cold atmospheric plasma for tailoring of surfaces, both for joining, but also for immobilization of bioreceptors. And then, of course, use of roll-to-roll fabrication with sustainable materials for volume manufacture. And also flexible, stretchable printed electronics into textile garments for wearables. So these were some of the key elements within the SusFE projects, the key innovative aspects, and they were built into a number of important use cases. And that's what we'll be focusing on today. So around next generation wound monitoring, blood cell sampling, a multimodal wearable platform for cardiometabolic monitoring, and a point of care diagnostics that's delivered in a very sustainable way. So we're going to have colleagues presenting on each one of these use cases, and as I mentioned, I'll ask them to introduce themselves. So the first use case is around next generation wound monitoring, and I'll ask Florian to introduce himself. Thank you, Florian.
Okay, thank you, Zulf. Good morning, everyone. So I'm Florian Dacricio, product manager at BFC. Unfortunately, our colleague Jamila Boudaden from Fraunhofer could not attend due to technical issues, so I will take over the presentation for the part Fraunhofer developed, and I'm happy to guide you through the development we've been conducting on the wound care use case for the SusFE project. Next slide, please. Okay, so as an introduction, let's start with 2 simple questions. The first one being, why smart wound monitoring will matter in the future? and why make those developments. So first of all, it will bring comfort to the patient and benefits, as it will enable post-surgical recovery at home with continuous passive monitoring, and overall reducing the patient anxiety. Chronic wound management will be transformed by allowing remote monitoring and also provide objective data, replacing the more subjective visual and olfactive assessments that are usually done on wound monitoring. Then it will give clinical and economical efficiency, as it will reduce, for example, unnecessary patch changes, and also free up some time to nurses and clinical professionals. And it will overall lower the cost of care. And so overall, it will have benefic impact on the healthcare system, as it will address a growing burden on this system. Next slide, please. The second question is about the strategic entry points. And for SusFE, we've chosen to focus first on a reduced set of simple and well-understood parameters that are clinically meaningful to the professionals. So basically, we've targeted temperature, as early wound temperature changes can indicate infection before visible symptoms appear. And also temperature sensors are compact, low power and low cost, and also easy to integrate. The humidity level, as an optimal wound moisture supports healing, and also a sudden increases in exudates may indicate an infection. So it's relatively interesting data to monitor. And the third one was pH, as wound pH shifts towards alkaline values can indicate a bacterial infection. pH sensors can be integrated into dressings, though the stability and the miniaturization remain the challenge. Next slide, please. So during the SusFE project, we've been developing two demonstrators. The first one being a temperature and humidity wound patch demonstrator, and the second one being a pH sensor demonstrator. So I will first start with the temperature and humidity demonstrator. In medical applications, such as smart wound dressings, disposable devices are treated as medical waste and are typically incinerated after use. Therefore, conventional batteries containing metals or hazardous materials are not best suited for those applications. The use of the biofuel cell developed by BFC as a power source eliminates the need of those conventional batteries. It is a metal-free energy source with low environmental impact, and therefore it is safer and more sustainable for disposable connected healthcare devices. And therefore it will also ease the end of life of these products. Next slide, please. So the enzyme stability has long been the barrier to the real-world applications of those biofuel cells. And BFC has developed a way to stabilize the enzyme in a carbon biopolymer matrix, that render possible to deliver days to multiple weeks performance for the use of those biofuel cells. The materials that we use are safe, as the biofuel cell is only composed of carbons, biopolymers, enzymes, and a bit of sugar to power the device. Basically, we transform glucose and oxygen into electricity. And so for this use case, we've decided to use the biofuel cell to showcase that we can actively activate such a demonstrator. Next slide, please. So what's the concept? It's rather simple. It's an assembly of a biofuel cell and a low-power electronic platform together within a medical-grade dressing. And as you can see on the picture, we can have a rather simple device that can be directly attached to the skin. It is composed of a biofuel cell, the biofuel cell having also a fuel and electrolyte reservoir that can be simply activated with a pull tab. And the electronic platform is composed of a deported temperature and humidity sensor to be able to measure the data at the heart of the wound. It is also composed of a real-time clock to monitor really the data on time, and a BLE and an NFC antenna to have the ability to communicate instantaneously the data. Next slide, please. So how does it work? Basically, we activate the device. So here, as a demonstrator, we use just a pipette to put the liquid, to pour the liquid in the cell. But as mentioned, we also have a blister to activate the device. So it is a water-based electrolyte containing sugar. You add the liquid, and then within a minute, you reach the SusFE, which is about 0.75 volts. Next slide, please. So we have a functional demonstrator to measure and transmit data via Bluetooth and NFC. And so here on the picture, you can see that we can use an app to retrieve the data and get the graph showing the variation of temperature over time. The system can operate in the ambient conditions, including ambient temperatures up to approximately 35 degrees and relative humidity levels from 15 to 85%. Next slide, please. So as mentioned, we can also get a continuous wireless communication via Bluetooth. And so, as you can see, one line is one data point, and then we can extract temperature and humidity variation over time. And the lifespan of such a device is approximately 5 days. And yeah, therefore, it's an easy to use simple patch. Next slide, please. So the second demonstrator is a pH sensor. Next slide. A novel field effect transistor concept was developed, which is suitable for chemical sensing in liquids, especially proton detection for pH sensing. It consists of two separate components, a capacitor with a control gate and integrated chemical sensitive layer. There are two readouts, a metal oxide semiconductor transistor, as well as an operational amplifier. A floating gate couples both components. The sensors are fully complementary metal oxide semiconductor, compatible, and have been fabricated on 200 millimeter silicon wafers, as you can see on the right corner. The chip size is about 20 square millimeter, which is a 4.5 by 4.5 millimeter device, containing 3 detector capacitances and 1 counter electrode. Contact pads and transistors are placed near the edge of the chip. This allows easy access for bonding, and by appropriate chip molding, transistors and amplifier can be protected from any disturbing effect of light. Next slide, please. So, as mentioned, we have 2 readout modes, one MOSFET and one operational amplificator, sorry. In SusFE, we used the MOSFET signal evaluation mode. The floating gate voltage is determined by VCG and the counter electrode voltage. Charges at the sensitive layer cause a potential change, so delta phi, and the floating gate voltage is changed by the floating gate voltage. Okay, drain current change is proportional to the floating gate potential change. Sorry. Next slide, please. The transfer characteristics of the readout MOSFET for pH values of 2 to 10 are shown on this slide. The reference voltage is kept constant and the control gate voltage is swiped between -5 and 5 volts. As expected, the threshold voltage shifts to higher gate voltage with increasing pH value, that is increasing proton concentration. For a constant drain current, we plotted the change of control gate voltage versus pH. Due to capacitor divider, the control gate voltage change is 115 millivolts per pH. Next slide, please. So some key outcomes from this part of the presentation. So on the temperature humidity wound monitoring demonstrator, we've demonstrated that a metal-free biofuel cell enables smart medical devices. The lifespan is about 5 days, depending on the number of measurements taken. A deported sensor has been implemented to measure the data as close to the wound as possible. And continuous wireless communication via Bluetooth and NFC render it easy to use and easy to implement. For the pH sensor demonstrator, silicon-based pH sensor enables pH sensing from 2 to 10. Drift in pH level is depending on the stability of the reference electrode. The lifespan observed is about 8 days. And designed for integration with flexible, it was designed for integration, with flexible substrates to be as easy as possible for wearable application. And that's it for the wound monitoring use case. Thank you.
Thank you very much, Florian. So as it was mentioned in the chat by Paula, if there are questions on that one wound monitoring use case, if you could please put those into the Q&A, that would be great. And our next speaker is Anna. So over to you, Anna. Thank you.
Thank you. So hello, everyone. My name is Anna Olander. I'm the R&D manager at Capitainer, and I will present the progress done on the blood self-sampling use case. Next slide, please. So I'll start by introducing you to the concept of microsampling. This is a new alternative to traditional venipuncture sampling, which is the gold standard way of taking a blood sample today. In venipuncture sampling, it requires that a patient travels to a healthcare facility for a blood draw done by a medical professional. Several milliliters of blood are collected in tubes that are sent with cool transportation to the lab for analysis. In microsampling, however, we are collecting smaller blood volumes, typically less than 100 microliters, and they can be collected through a simple finger prick and stored in a dedicated microsampling device and shipped to the lab for analysis via regular mail. So this enables self-sampling, that a patient can take a sample by themselves in their homes, and it makes blood testing more accessible and convenient. It can lower cost for the healthcare system, and there's also an environmental aspect in terms of reduction of the amount of transport, so we can lower the carbon dioxide footprint. Next slide, please. So, thank you. So in the SusFE project, we aim to develop a self-sampling device with an integrated timestamp. One back, please. So, yeah, there. The reason for including a timestamp is that in some healthcare applications, knowing the time when a sample was taken is essential. Recording patient ID and sampling time is standard practice when you take a blood sample. But in therapeutic drug monitoring, where drug concentrations are measured at specific time points, you need very precise measurements, and you do this to be able to evaluate drug efficacy and to avoid toxicity. So this process is widely used in pharmaceutical drug development in the pharmaceutical industry, and also in healthcare. And as pharmaceutical industry is moving towards so-called decentralized clinical trials, where patients or the study participants remain in their home and are monitored remotely instead of going into a trial site for checkups and testing, there is a need to be able to do TDM through self-sampling. And there are studies that have shown that, of course, you can do self-reporting of sampling time and other parameters, but recordings often happens incorrectly or missed out entirely, which can lead to significant data loss in clinical trials. So this is the reason for us to integrate a timestamp in the device. Next slide. So in the SusFE project, we did market analysis and use case analysis, and we identified 2 primary users, and it is the pharmaceutical company doing clinical trials and are striving toward decentralized clinical trials. And it's also the healthcare provider that has patients that rely on drug monitoring. For example, a kidney transplant patient needs to monitor their drug levels throughout life. So we interviewed representatives from these user groups and to help define key product specification, and you see some of them here to the right. The device should produce plasma, not whole blood, because gold standard sample matrix for drug development is plasma. The timestamp must be passively activated, meaning the user should not have to push any buttons or scan QR codes to activate the data collection, the timestamp collection. This is to reduce the risk of data loss. We need about 1 to 2 minutes of resolution in time, and the time should be able to be recorded for a minimum 5 days to be able to ensure that you have your recording when you ship your sample through regular mail. And when the sample arrives at the laboratory for analysis, you should be able to wirelessly read out the gathered data. Next slide. So with these specifications, we developed a device concept that considered meeting these product specifications. And we want to do this using sustainable electronics components and also a scalable manufacturing process. And here you can see an overview of the partners involved in this use case and the building blocks they bring in the form of their proprietary technologies. At the core of the device, we have the Capitainer volumetric plasma sampling technology, and we set out to integrate carbon-printed electrodes in the Capitainer in this plasma sampling microfluidics that then can sense the presence of plasma in the system. And here we use VTT's printed electronic technologies. The signal from these carbon-printed electrodes will be picked up by a flexible integrated circuit from the partner Pragmatic. And this Flex IC also contains the technology for wireless readout through an NFC. And it all will be powered by the bioinformatic fuel cell from BSE. And to put it all together and manufacture it, we will use VTT's roll-to-roll production line. Next slide. Okay, so let's have a look at what we achieved in the end after these 3.5 years of collaboration and development. As a starting point for the plasma device development, Capitainer had the technology that was available on a prototype level. And a unique feature with the Capitainer plasma sampling technology is that it can carry out plasma extraction and metering into an exact volume completely passively. So the user just applies blood to the system, waits a few minutes, and then you have your sample. And during the course of the SusFE project, Capitainer transferred the technology to production, and the product is now available as a CE-marked product, CE-marked according to IVDR. And here you see an example of device performance for ferritin measurement. Ferritin is the hemolysis sensitive biomarker, so you can't measure it in whole blood. And we see that the volumetric performance of the device is very good. We end up with a method CV below 10%. So this is an important aspect for the future exploitation of the SusFE device, that we have a good plasma sampling technology. And move on to the next slide, please. And so this CMOT product is called Capitainer 10. It's currently being produced using manual assembly. It's based on plastic foil-based construction, so it's suitable for roll-to-roll manufacturing. And in SusFE, we developed a scalable roll-to-roll manufacturing process for printing of these activation electrodes, and also for manufacturing of the entire microfluidic components. And here on top, you see ETT's roll-to-roll production line, where we have printed electrons in meters of foil. And on the bottom, you see how these electrodes are integrated into the microfluidics. We've worked both with roll-to-sheet and also the final goal, we enable to make a complete roll-to-roll process. You see that to the bottom, to the right, electrodes with some microfluidic layers. Next slide. And we also tested the functionality of the electrodes and the microfluidics. And here you see the assembly. We have microfluidics and integrated electrodes, and here we have added blood to the system and connected the electrodes to an LCR meter to measure capacitance. So to the right, you see how the capacitance of the first electrode increases when plasma enters the plasma metering channel. And then there is a second increase later when plasma enters the second metering channel. So we can clearly measure a clear response change in capacitance. And so the concept of printing these carbon electrodes on a foil layer adjacent to the plasma metering channel is viable. Next slide, please. So we have a way to detect that there is plasma in the system, and this signal needed to be picked up by the Flex IC to start the time counting. For this, Pragmatic designed and manufactured a dedicated Flex IC, and we also developed a process for how to integrate it into the microfluidic components and how to connect it with the electrodes. And here, to the top right, you see the resulting Flex IC component that was produced. But unfortunately, we were not able to get the Flex IC to function together with the other components. And this is highly likely due to the fact that Pragmatic needed to deviate from their standard manufacturing process for this use case. And we didn't have time or budget to solve this issue within the project and make a new production run. So this is something that we have to look at in the future. But nevertheless, on the video below, you can see how the component is working outside of the blood sampling use case. You see how it picks up and transmits as timestamp to the smartphone. Next slide, please. Then we come to the biofuel cell, and BFC designed and manufactured a biofuel cell for the use case. And as an intermediate solution, since we didn't have a Flex IC that we could integrate, they also designed an electronic platform using conventional flexible PCB. However, we were not able, due to the budget and time constraint, to develop the platform with a timestamp. So here we were able to borrow some features from the wound monitoring use case to monitor temperature and humidity instead. So these parameters, temperature and humidity, were not part of the original specification, but they are parameters that are highly interesting and relevant for the use case. So it will be integrated in the future product. You need to be able to control or analyze stability during the shipment. So temperature and humidity monitoring is highly relevant. So the biofuel cell needs activation through access to the electrolyte. And to enable this, BFC designed a concept using a blister pull tab. You see it at the bottom left picture. And with this pull tab activation, the fuel cell can be integrated into the packaging of the plasma device, so activation actually can occur passively. And again, passive functionality activation is a very important feature for this use case. To the right, you see some characterization data. In this configuration, the device can run from ambient to 35 degrees Celsius. The lifespan is about 2-3 days, looking at the biofuel cell tension, and with a small decrease at higher temperatures and low relative humidity. And the data can be extracted via NFC, you see it at the bottom, and with this, the lifespan is confirmed. Next slide, please. We also, so we have our components, but we also needed a way to put them together. So we developed a scalable manufacturing process for final assembly of the components. And here you see the VTT Yaskawa pick-and-place robotics being used to pick up the biofuel cell and place it on top of the microfluidics. That's under A. You see microfluidics and fuel cell, and then how they are put together. And how this robotics works is that they can be programmed, and then you scan a QR code to recognize which component it is and where to place it. So below you see how we have used this pick and place process to put together the different components. And then for electrical connection between components, we can use standard electrically conductive tapes or UV curable conductive adhesives. Next slide, please. So that was automated assembly, but we also wanted to put the components together for final functioning demonstrator. So that's what you're looking at here. We put them all together manually and tested them. And here you see our final SusFE blood sampling demonstrator. We have on the picture to the left, we have the microfluidic component with the integrated activation electrodes produced by VTT. And here it is assembled in the Capitainer SEP10 packaging. On the lid of the sampling card, we have assembled the fuel cell and the electronic platform. And this demonstrator could extract plasma, and the electrodes responded to the presence of plasma in the channel. And since we were not able to fix the issue with the FlexSE, we were not able to demonstrate the timestamp function on chip. Instead, we are demonstrating temperature and humidity recording. You see it captured here through NFC on the smartphone and the picture to the right. So we're not able to integrate timestamp function in this platform, but adjustment of the platform to capture timestamp is rather straightforward engineering. We just didn't have time or budget within the SusFE project. Next slide, please. So to summarize, some key outcomes: a microsampling device with integrated functionalities for capturing of sampling time and recording of shipping temperature and humidity was developed. Passive timestamp activation was achieved. We could produce the system using roll-to-roll. We have realized a biofuel cell that could power timestamp, temperature, and humidity monitoring based on silicon integrated circuits. And we did realize a FlexSE that can initiate a timestamp triggered by a change in capacitance. And the next step for us is to find further financing to take the technology towards commercialization. Next slide, please. And that's my final slide. I just want to close by highlighting what makes this future product unique and how it compares against existing solutions. So our market analysis showed that there are currently no commercially available microsampling devices with an integrated timestamp. We found a few prototypes presented in publications and on trade fairs, and if we compare ourselves to these, our system has some unique features, and that is that it collects a volumetric sample and enables quantitative analysis, and it does so passively without requiring user intervention. The timestamp is activated passively, other systems that we have seen needs to be initiated by you. You need to push a button or scan a barcode to register the time sampling. And our system uses sustainable electronics. All other prototypes or systems that we have seen use conventional electronics. And finally, we can also compare it, the SusFE demonstrator or SusFE concept with the gold standard way of doing TDM venipuncture sampling using a manual registration of sampling time. And here the integrated passive timestamp supports increased robustness in sampling, time data collection, and minimizes data loss, because in our interviews, even nurses can do a wrong registration of time. And finally, we were able to put some numbers to how much our system reduces the amount of CO2 emission, with referencing a publication from Johnson & Johnson, who did life cycle analysis on their trials, and it's estimated to be 1,000-fold reduction using self-sampling with integrated timestamp in clinical trials. So it's quite substantial.
That's great. Well, thank you very much, Anna. It's really interesting. Okay. Oops. So, Mattia, over to you.
Thank you, Zulf, and thanks to everybody attending. It's a pleasure to be here. I'm Matija Strbac. I represent Technalia Serbia, which is basically a technology accelerator office of Technalia Research and Innovation based in Spain. And we have been working now for more than 18 years as a technology accelerator office, meaning that we are focusing on fast cycle development project and prototyping devices in biomedical applications. And thereby we have worked primarily on basically wearables, electrodes and sensors, wearable electronics, in the domain of neurorehabilitation, but then also in the domains of anything that can cover basically the data acquisition, electrophysiological assessment, etc. And hereby my talk today will be basically how this all converges into a cardiometabolic wearable platform that we have now developed in the SusFE project. Next slide, please, Wolf. So basically the concept what we have developed prior to starting the project is to design a next generation of cardiometabolic wearable devices. Why? Because we believe now is a great timing, meaning that in the technological perspective, these electrodes and sensors that we were developing for years now are allowing basically multimodal sensing, coupling electrochemical and electrophysiological measurements into the wearable devices. And then there is also this other point that, now today, machine learning and AI using this multimodal signals data, multiple signals data fusion is now allowing us to assess some information much better. Our focus here was basically to evaluate physiological load and psychological load. And we believe that there are plenty of applications of these technologies. We have designed this as basically multimodal system, meaning that it's also something that is basically modular and is a platform. So all of these different signals that we can now convey and basically use through this platform will allow us a wide range of applications, which I will comment on later. Next slide, please, Ulf. So when we look at what is the current status and where we see competition for such a device, of course, obvious thing that comes to mind is these typical smartwatches that now is, there are plenty of them all over the market, right? And people are now, basically switching from conventional watches to smartwatches more and more. Of course, younger generations are coming. This will not stop. And people are using this more and more for assessment of general health and fitness. But this is, of course, rudimental type of measurements. They're usually, let's say, using 2 to 3 different signals. For instance, speaking about the heart rate and ECG, they are usually focusing on plethysmography-based assessment. And of course, there is a plenty of variability in this. And like I said, number of signals and the potential of the data fusion and the machine learning, which is already part, of course, of the regular smartwatches, is in that sense limited. Of course, there is now also advanced smartwatches that are used for better assessment, let's say, but there is these are still not having any biochemical sensing and other capabilities that I will introduce later. And then, of course, there is something that we see as, let's say, clinical halter systems, which are now having a tendency to somehow converge to simplify versions with less electrodes, but then at the same time they are losing the resolution of the signal, and of course there is a clear trade-off there. And we would like somehow to have this as a multimodal system to be able to keep as much as data as possible. Of course, last but not the least, there is plenty of application of such system in sports and smart garments and use in basically recreational but also professional sports. Next slide, please. So how we have envisioned the system is basically that we have two key components. One component is basically this electronic device, box enclosure, something that is very close to the conventional devices that you're used as mobile phone, well, a little bit smaller than mobile phone nowadays, but mobile phones from 20 years ago, I guess, in terms of the size. And then there is this other part, which is for us also very relevant, is this printed electronics basically part that is a wearable, that where our focus was basically to make this paper based, to make this as a biodegradable part of the system that is leaving basically minimal waste. And this is how the system was envisioned in terms of the components. Next slide, please. But in terms of what are the goals for us in terms of the sensing, we wanted to couple physiological and autonomic signals basically coming from ECG. We want to measure ECG as mentioned before, not through plethysmography, but basically through ECG electrodes, of course, with interesting placement of the system. Then electrodermal activity, because we want to couple both, and the temperature as well. We have the other part that is, of course, a little bit conventional. It's IMU, but considering that we want to couple this information, and for us, this is very important, the placement of the electrodes and the measurement that we know exact, well, basically kinematic data. And then the third part, which is let's say the most ambitious one, is the part that is related to biochemical measurements, where we have integrated the cyclic voltammetry, amperometry, but then also couple this with antiphoretic extraction, and the part that is related to body composition analysis or bioimpedance. So all of this is integrated into our system that you can see here. The electronics is supporting measurement of all this data. And then next slide, please. We have also introduced all of these means of measurement of this and interfacing the human in our multimodal electrodes. I will not go into the details here, but you can see that there on the electrodes, there are several things that are relevant. There are the parts that are electrochemical electrodes that are in the middle of the circular top part, but then there are ECG electrodes all around, and there is one temperature sensor, which we have used also special type of inks for that are thermally sensitive to measure this. But for us, most important part is basically this modularity of the design. What we have done is basically we have split the part that is the leads for the electrodes that are for us integrated into the garment, and that we consider basically something that will be used over time, but is also allowing washability. But on the other hand, the part that is sensors, this is something that we consider basically consumable with the sensors and the electrodes. This is printed on paper with the minimal use or actually non-use of silver elements. Everything is carbon and PSS based, and this is the electrode that you can see here. Next slide, please. Now, when we have all these components, basically physical components, means of interfacing the human and gathering the data, here you can see how the sensor is placed on on the subject. You also saw this basically in the concept image on the first slide, but here you can also see the Bluetooth communication in the Android device, and we have developed the Android application basically that is allowing us to control the device and make all of these different measurements that I have introduced, and also allowed it to do this synchronously. So basically for us it was important to measure the IMU data coupled with the ECG data coupled with the data from of GSR, because these all events are, let's say, correlated, and for us it's important to use sensory data fusion to do a certain assessment. Now I'll speak about what we are actually doing. Next slide, please. So basically you can see that there is of course some pre-processing that needs to be done. You can see on the top left side, you can see the ECG, how the raw signal looks like. From the raw ECG, of course, there is a movement artifact, there is a baseline drift, there are parts that is, well, clearly noise, 50 hertz noise, right? And then, of course, with the right approach of filtering, we can come to the signal that you see there next to it, basically, and we are able to annotate directly through this algorithm the R peaks, basically, and to estimate the heart rate. Once again, this is this important differentiation. Basically, here you can see the ECG data. This is not the same as you can get through, let's say, modern smart watches in terms of PPG. This is allowing much more information, but of course, for the assessment that we are doing here, we are using specific features of heart rate. Then the other part that we are also measuring is GSR. Here you can see on the bottom graph how this signal looks like during the raw measurement, but for the assessment later, we actually need to filter this to get the two important components of GSR for assessment of stress. One is the tonic and the other is fuzzy component. And you can see on the, well, let's say central top image, you can see how this signal after filtering looks like. And then basically based on these two signals after the pre-processing, we are able to come to the features. The important heart rate features and important features of electrodermal activity or GSR. You can see the list here, but for instance, basically in our example, there is 20 features that we have chosen, and there is of course the temperature sensor as well. I will not go through the list, but this is some of the, let's say, key components and key features of these two signals. When we have this, we can actually now start playing with the machine learning and AI. Next slide, please. So basically what we have done is we wanted to validate whether through these signals we are able to assess the stress. And we have done an experiment on the assessing of cognitive or physiological load, basically that is called MIS. This is a standardized test for introducing higher levels of stress. Zolf, maybe you can also play a video. It's not really interactive yet, good, but it's actually showing how the test is going. So subject is actually doing some arithmetic task. He's asked basically to add and subtract certain numbers, and he has very little time to provide the answer. And then this timing is over time being reduced, but there are also different stressors that are introduced. One is actually a comparison. Subject is told that there is a level of other subjects, and basically he sees that he is getting bad in comparison to the others. There is also time limit that I mentioned. There are verbal comments that the interviewer or basically the experimenter is giving. And then there is also a video recording. So basically this is introducing different levels of stress through the experiment. I will not go in details here, but idea here is that this allowed us to measure the data on 20 subjects and basically assess what we are able to how precisely we are able to assess the stress through these signals. Next slide, please. And here you can see, of course, that this experiment is not ideal in the sense that, of course, you cannot really control what is the level of stress that you will introduce. Maybe subject came stressed at the start of the test. Maybe you are trying to increase the stress levels, but maybe it's not possible. Some subjects are also not so, let's say, easily provoked. And here you can see on the left side, basically the subjective stress score. I wanted to show you this just so you can understand that actually what we are here targeting is not exact numbers. We are not really able to say that each of these phases that we had, 4 phases during the experiment, will ideally fit into the 4 levels of stress. But what we wanted to do is, let's say that to do it as precisely as possible, we have assessed that each of these phases is representing higher and higher level of stress. And what is interesting here to see, and this is why I'm actually focusing on this slide, is that actually when coupling this data, of course, you are able to assess the stress only from the heart rate data. But when you mix heart rate and electrodermal activity, you're getting much more precise in the assessment. And here you can see the comparison and in this Weill plots in the center. But what is also even more important is that actually you can easily say that the electrodermal activity is much better for assessment of stress at higher levels of stress. And here you can see in the bottom table actually how the numbers are. Of course, I'm not going to the details here, but actually this slide here was to convince you that the coupling of data and the machine learning that we have provided inside of the system is allowing us to get much better assessment of stress. Next slide, please. And this is my final slide. I wanted to explain here that basically the system that we have designed is designed as a platform. And what we have already recognized is that different components and this system is modular, so different models of this system will allow the flexibility in using it in various applications. So there is, of course, in research and in clinics, this can be used for physiological monitoring, for stress and health studies. Of course, I already mentioned this was not in the, let's say, AI experiment that I showed previously, but this is now system is allowing this to be coupled with electrochemical assessment. So with different biomarkers, you can get even better assessment of specific states that you're targeting for. Of course, it can be used also for everyday stress monitoring. Clearly, it will give you a little bit more advanced information in comparison to well, let's say nowadays smartwatches. And then, of course, these smartwatches are being used more and more, so clearly there will be an interest for that as well. But where we see, and for what for us we believe is the next step of the evolution of such a system is in this Worker 5.0 applications. And hereby I would like to say that we actually plan to continue this through another European project that will start in September, that's called Growth, where we will try to focus this technology towards the use of the future factories and to couple it with collaborative robots to allow the assessment of the, let's say, not only single out workers, but basically the factory, and to optimize the human-robot collaboration with such technology. There is, of course, use of this type of system in sports and fitness, as mentioned previously. Healthcare providers, patient monitoring. So basically, this clearly is something that is now this telerehabilitation and the parts that is related to basically telemonitoring of patients clearly is providing this type of technology would improve. And last but not the least, wellness and lifestyle in general, this type of technology is something that will be a mediator. So this is all from my talk, and I'm looking forward for some questions that you will hopefully write into the questions and answers chat box. Thank you all.
Okay, thank you very much, Mattia. And yes, just a reminder, if you could please put your questions into the Q&A and we'll take those at the end. And so great if I can invite then Anu and Mariam to talk about the final case study that we developed, which was around the point of care diagnostics. Thank you. Over to you, Anu and Mariam.
Thank you, Sulf. Hello, everyone. So my name is Anu Mursula, and I work as a senior scientist and project manager at VTT, Technical Research Center of Finland. And I'm here with colleague Mariam. Please introduce yourself.
Yeah, hello everyone. I'm Mariam, a senior scientist at MPG, and I'm very glad to briefly describe how our cold atmospheric plasma technology has been applied in SusFE project.
Okay, thanks, Mariam. So Mariam and I will present the point of care diagnostics use case and how sustainability was taken into account in the development of this use case and how technologies from different partners were integrated into the whole thing. But first, some background about point-of-care diagnostics. So point-of-care diagnostics refers to medical in vitro diagnostics, testing that is performed at or near the site of the patient care. So instead of sending samples to traditional central laboratories and waiting hours or days for results, point-of-care testing delivers results usually within some minutes, at least less than an hour. And generally, the need for diagnostics in healthcare is constantly increasing, for example, due to aging population and the increasing prevalence of infections and chronic diseases. Also, the interest of people for self-testing is increasing the demand of these tests. Point-of-care diagnostics is so relevant in healthcare today because it brings, as I mentioned, the testing closer to the patient and speeds up the result, and because of this, can improve the outcomes. In addition, it enables monitoring even at home. A very common example for this is the glucose monitoring in diabetes management. At the same time, the growing use also creates a sustainability challenge. Many of these point-of-care tests are single-use tests, and they generate significant amount of waste, which was clearly seen during the COVID-19 epidemic. So especially the plastics waste is increasing due to this a lot. So the plastics comes in different forms: the cassettes, foils, lancets, buffer tubes, swabs, packaging. And sometimes we also have single-use electronics as waste in these tests. So next we'll have a look a bit more closely how SusFE was trying to address this sustainability challenge. So here are summarized the key requirements for the use case and the goals of our development work. We can highlight two main dimensions: the sensing approach and target analytes on one side, and sustainability plus scalable manufacturing on the other. In practice, this means that we need a sensor platform that can detect clinically relevant biomarkers reliably, while also being compatible with sustainable materials and high volume manufacturing methods, like roll-to-roll manufacturing. So in this use case, we decided to focus on electrochemical sensing as the technology platform, and by introducing either specific antibodies or aptamers on the surface of the sensor to capture the specific biomarkers. And we are targeting two important biomarkers in this work. One is cardiac biomarker troponin I, and the other is inflammation biomarker CRP. In the results section, however, I will focus on the aptamer-based troponin I sensing because it showed more promising results than the antibody-based CRP detection. For the readout of the electrochemical signal, results were used for the readout. We used a commercial potentiostat, commonly used in the laboratories for reference, but in addition to this, we also developed a small handheld integrated circuit potentiostat, very small one. And we also investigated the possibility to design integrated electronics, such as flex IC to the device. Next slide, please. So on this slide, you can see the contributing partners and the specific roles that they play in the point-of-care diagnostics use case. So VTT was the use case leader here and was responsible for the overall design of the point-of-care device and its fabrication by using sustainable materials. In addition, VTT also investigated possibilities to functionalize the sensor with CRP-specific antibodies for the electrochemical detection of CRP in samples. Molecular Plasma Group MBG provided plasma spot technology for Teesside University to enable plasma-assisted immobilization of a linker layer on the sensors. After the linker immobilization, Teesside University continued with the troponin-specific aptamer binding to the linkers, followed by troponin detection. Teesside also developed the handheld measurement device for differential pulse voltammetry measurements from the point-of-care sensors. This device can be used thousands of times, while the point-of-care sensors themselves are single use. As an alternative for this system, we investigated with pragmatic semiconductor the possibility to develop an integrated electronics and flex IC to the system, as you can see from the bottom left corner. However, this is a concept only, not a functional prototype yet, so that would require a bit more work. Next slide, please. Here I'm showing how we use sustainable materials in the point-of-care diagnostics use case. The key idea is that the large share of the device mass, so totally 81%, was made from bio-based materials, which shows that more sustainable design is possible even in technical diagnostics applications. Basically, we replaced conventional components, mainly plastics, where possible, with bio-based materials without losing functionality. For the base material, the so-called substrate, we replaced the conventional PET plastic with polylactic acid, PLA. The electrochemical sensor elements were printed on the PLA by using mainly carbon graphene inks. And silver chloride ink was used only for the reference electrode for better stability. For the insulator layer, a UV curable commercial material was used, although also beeswax was briefly investigated as a bio-based option. For the microfluidics layer, a conventional double-sided adhesive was used, but the lid layer again was possible to be fabricated by using a sustainable material, in this case, cellulose acetate. Then we still had the blood filters and absorption baths to finalize the test chip, and there we used mainly cellulose material, but also glass fiber as materials, but the mass of these is quite minimal. So on the next slide, we see how the point-of-care diagnostic chip was then manufactured. The sustainable diagnostics need to be designed not only for performance, but also for how they can be produced efficiently at scale. For the printing of the electrochemical sensor on PLA, roll-to-roll printing line at VTT was utilized, as you can see on the left. This enables printing of a large number of sensors in relatively short time. For the microfluidics part, including the lid, we fabricated it by laser cutting at VTT on a sheet scale process, but this can be easily transformed to a roll-to-roll process as well. Before the integration of these elements together, there needs to be the biofunctionalization step on the sensors to make the sensors capable of detecting a specific biomarker. At the moment, the biofunctionalization process is still quite time-consuming and a multi-step process that would need to be some further developed to be a large-scale process. But in this project, we were able to do it in lab scale very nicely. And finally, the sensors and microfluidics and the filters are integrated together, followed by cutting into single test chips. And then we have characterized our sensor elements for the repeatability and quality before any immobilization of aptamers antibodies. And we characterized these by differential pulse voltammetry, DPV, and also cyclic voltammetry, and compared the results to commercial electrochemical sensors. The peak DPV currents of the VTT sensors, both on PLA and PET. So here I need to mention that we also printed the sensors on conventional PET for comparison. And both of them had very repeatable results, but due to the use of carbon in the electrodes instead of silver, the peak currents were lower than compared to the commercial electrodes. So this is a slight challenge for the actual detection of the biomarkers, because the signal level is pretty low in the starting point, and then the small changes in the signal levels due to the biomarker binding might be a bit more challenging to see. But on the positive side, the VTT sensors had lower variation than the commercial sensors. And we could see this also from the bigger analysis batch than seen on this slide. So we have analyzed more than 100 sensors before, and the repeatability of our self-made sensors was very good. So next, we'll see how the modified sensor surface of the troponin eye detection will be performed, and Mariam will continue here.
Thank you, Anu. So first, I would like to emphasize that all work shown here was performed by TSAT University using our plasma spot technology. So to avoid any confusion with blood plasma that Anna interestingly explained for us earlier, I briefly explain about our plasma technology, which uses an aerosol-assisted plasma deposition in DBD configuration. So it generates plasma, ionized gas, in atmospheric pressure without any arcing. It means that it's glow discharge, and at room temperature. So maybe Sylv, if please play the video, the other one. Yes. So the precursor injected in the afterglow. So it's very key role here, because it preserves the sensitive of organic chemistry and then sensitive substrate, so it can be easily functionalized without any damaging. So as you can see, it's the comparison between our technology with the other technology available in the market. And when I say that is aerosol assisted, so maybe next video. It means that first we make aerosol from the chemistry that we intend to functionalize our surface with, and then we introduce it in afterglow, so it preserves all of those functionalities. So overall, our plasma is very gentle and provide control the environment to functionalize any surfaces, even the sensitive ones. And also almost all organic chemistry can be injected and functionalized with our technology. So next slide. So let's see now how our plasma technology make changes in SusFE project. So at the first, the carbon electrode the surface of carbon electrode was tailored using argon plasma. TSAT University activate the carbon electrode first, as you can see in the STEM pictures. If you compare before and after A and B of plasma tailoring, plasma treatment, you can see that the roughness of the carbon electrode is increased. Also, the porosity is higher. So So also the wettability of the carbon surface is now higher, so it's make it for the next step. And interestingly, you can see that how this first step of plasma tailoring, plasma treatment impacts on the electrochemical results. So there is a promising improvement in the charge transfer efficiency, as you can see in EIS test. So now our electrode is ready for next step. There is a point that need to be mentioned. For bioimmobilization, we need a linker layer, which is typically carboxylic acid groups. And normally, typically, they provided by wet chemistry. And it consists of multi-step and also high level of solvent and also energy. And as Anu mentioned earlier, one of the key objectives in SusFE is sustainability and green solution. So it's again where the cold atmospheric plasma changed the game, and then within one step, solvent-free and dry procedure, it provides a high level of carboxylic acid functional group and make those active sites for the next step of optimal immobilization. So what is important here to mention is that the plasma activates the surface as well as the precursor simultaneously. So it's the reason that this carboxylic acid group covalently grafted to the substrate and make it prepare to be also covalently grafted to biomolecule, which is optimer here, and give us a stable optosensor, which result will be presented again with my colleague Anu. So the floor is yours. Thank you.
Thank you, Mariam. So now we have a sensor with specific aptamers attached to them, and they are ready to bind the biomarker of interest. And in this case, it's troponin I. And here we have now tested these sensors with various concentrations of troponin I, both in buffer and in serum, and made calibration plots of this concentration series. And what you see here is that while you increase the concentration of troponin I, the peak current of the DPV curve is getting lower and lower. So this indicates that the sensor surface is having a blocking effect by the binding troponin I, which then leads to lowered measured current. And as you can see here, the lowering of the peak current was concentration dependent. So the calibration plots seem very nicely linear. And for the buffer-based samples, the limit of detection was 25.2 picograms per milliliter, and for the serum-based, 41 picograms per milliliter. So this is already a very, very sensitive sensor and very nice starting point for further development. So next slide, please. Yes, so furthermore, we developed this small integrated circuit potentiostat that can be paired with the APTA sensor. And this is capable of measuring DPV. So we compared the measurement performance of our reader electronics against the Autalab commercial reference instrument to see how our small and compact approach can still deliver reliable results. And yes, it does. And IC potentiostat gives comparable DPV results, which is a very important result, as you can see in the figures on the right side. And then finally, when the system is fully integrated also with the microfluidics and measured, we can see that the integration of microfluidics affects the signal levels somewhat, so the signal is lower. So this is a very common observation that you see in fully integrated systems. So the whole system needs to be taken into consideration when we evaluate the performance. And this is a very, very good starting point for further development. So I think on the next slide, I have still a few points that where I want to highlight the sustainability aspects and why this is unique, at least somewhat. So currently, there are limited point-of-care electrochemical sensors on the market that would measure protein analytes such as inflammation or cardiac biomarkers, despite of very heavy research ongoing on the field. So this indicates that the commercialization is quite difficult. But the technologies developed here in this project will contribute in the market entry for new possibilities for electrochemical sensors. And for the sustainability aspects, we were evaluating more sustainable material choices for the fabrication, and the plasma functionalization enabled sustainable, primerless, solvent-free surface modification that reduces chemical use and environmental impacts. And point-of-care diagnostics as a whole saves CO2 because you don't need to travel to healthcare facility and you can do the testing at home. And on the final slide, I have the key outcomes. So we were able to manufacture repeatable electrochemical sensors on bio-based PLA by using mostly sustainable inks. And in the microfluidics part, also bio-based cellulose acetate was used. 81% of the total mass of the device was made of bio-based materials, and promising concentration-dependent aptamer-based troponin I measurement results were obtained. Furthermore, the point-of-care chip could be paired with the small IC potentiostat. Comparable measurements results to commercial readers. So as a summary, we demonstrated a pathway for aptamer-based sensing using cold atmospheric plasma immobilization integrated in a microfluidic device with IC potentiostat, and this can be applicable for detection of a variety of biomarkers.
That's great. Thank you, Anu and Mariam. So that concludes our presentations, and we've now got some time for questions. So Paula, have we got any questions in the Q&A that we want to raise?
Yeah, we have one question from Victor. He's asking if the wound monitoring devices are applied on the wound or elsewhere on the patient's body.
Florian, do you want to take that?
Yeah, and that's a good question. So maybe if you can just come back to slide 12, could be easier to show directly. But basically, the prototype was designed so that the main part of the device is on healthy tissue, and only the sensor is really at the heart of the wound, and it's yes, maybe that one. Oh, so 11 maybe.
Sorry, let me just
yeah, just slide 11.
I beg your pardon. Thank you.
No, that's okay. Okay, so yeah, here you can see that most of the device is really can be on the healthy tissue, and only the deported sensor will be at the heart of the wound. And yeah, as I was mentioning, it's really interesting for biocompatibility aspects. We've been conducting some early assessments, and this design is quite interesting because only the sensor part will have to support the biocompatibility studies, as this is the only part that will be at the heart of the wound.
Okay, thank you, Florian. Victor, if there's any other points that you want to raise from that please do so. If I can just ask Florian, so what are the next steps in this development? So you've said that you've done some initial studies on this. So yeah.
Next step will be to push the study on the temperature and the humidity assessment to ensure that the device can last what the market need. And then we know that we need to work on the activation parts, because right now the blister is not really the best way to activate a patch. Actually, the blister was initially developed mainly for logistic loggers and trackers, which is okay for that application, but for a health patch, it's not really practical. So we are now working on that part to have an easy activation as a patch.
Yeah, great. Well, thank you, Florian. Actually, just on that, if I can, while people are thinking about any questions that they may have, if I can ask Anna, you mentioned that you've got one of the devices through SusFE has now gone into production, or it's CE marked and production. So I wonder if you could just say a little bit more about practically implementing some of these results, because you showed how you've got the carbon electrodes and integrating with the biofuel cell. So what are the next steps for you in terms of actually commercializing the outcomes from the project?
Absolutely. Yes. So of course, finding funding to be able to take that's the deciding point to be able to continue. And some of the partners in SusFE, together with VTT and Captainer and VTT, are applying for another grant where we will subcontract BFC. And we've managed to engage a partner from one of the big pharmas to join us as end user. As end user, and be part of specifications and testing what we develop. But it's quite substantial, a lot of work to get going from where we are. We're judging that we are at TRL 4 now, and to get it up to a functioning product. And there's also the regulatory aspect of it. Will it be, I mean, if it's going, well, it needs to be CE marked according to IVDR, and that's the question of what type of classification it will be. So, but we are very motivated to continue, and it will be Captainer that commercializes this future product. So, yeah.
Okay.
And concern what we learned in SusFE was a very important. We did a lot of the development using laser technology, which we have done together with VTT, but the transfer to production was carried out outside of the SusFE project, and there the layers or the component of the microfigures are die cut. So it was a really great learning, things to consider, how you work in development and how it will affect performance when you transfer to production and use different technologies. So that was a great learning for Captainer in working with SusFE project and to work with microfluidics.
Great. Well, thank you very much, Anna. So while we have you here, Anu, I just wondered if you could say a little bit around the I think it's a great point that you made around the waste that's created in lateral flow devices, and the potential for reducing that with the roll-to-roll manufacturing, different types of electrode materials. So if you could perhaps just talk a little bit more around how you see this developing for the future, and also a little bit around the bioreceptors, because that's always a sensitive part, and what the differences are between, say, antibodies or aptamers as well. Thank you. Or what you found.
Yeah. So we did some literature research on the waste problem, and I'm sure everyone has seen, for example, lateral flow tests. They are always in these plastic packages, and it's might be some 3 grams or so plastic waste per test, and it is just discarded. And then many of the automated tests that are done in laboratories might come in pretty bulky plastic cartridges containing all kinds of chemicals and different kind of parts, and these are also just discarded. So at the moment, we don't see so much sustainability being taught in the diagnostics industry, because the main thing, of course, is the patient safety and the performance of the devices. That is the most important thing. But I have seen some companies that are starting to raise the question that could we maybe replace some of the materials in these test devices with more sustainable options, or even leave it out completely. So is it necessary to pack all the, for example, lateral flow tests into plastic cassettes? I don't think so, not always. So yeah, I think it's a question that needs to be taken more seriously in the future. And concerning the bioreceptors in immunological tests and also antibody-based electrochemical tests and aptamer-based, the bioreceptor itself is the key thing. It's the most important thing, that it needs to be very specific, and the affinities needs to be just right. So I'm sure this will continue to be, of course, expensive and the most critical thing in the development. And in basic immunological tests, the implementation of these bioreceptors is quite well established. But as we saw in this project, the application in electrochemical sensing will be a big thing to solve in the next few years for the community. So
Yeah.
there are not many products on the market.
Yeah, I guess, lateral flow devices are qualitative, aren't they? And when you introduce a transducer, electrochemical, it really gives you the prospect of giving some quantitative information. And so you've got the potential there in terms of both reducing the waste potentially, and then also giving a more quantitative performance, or, raising the performance of the test as well.
Yes. Of course, lateral flow assay development makes also progressing towards more quantitative tests. But yeah, of course, quantitative, you always would prefer quantitative, I think.
Sure.
Yeah.
Great. Well, with that, can I thank all of the presenters for some really interesting presentations. I'd also like to thank colleagues from Medtronic in terms of organizing the webinars, and of course thank all of you for attending and listening to that.